Good morning, dear students. Today we are going to talk about two main points in genetics, which are sex determination in human and the abnormal genetic cases in human too. Let's discuss the first point together. In the past, it was thought that the female is the one who can control the type or sex of the fetus. But after discovering the sex chromosomes, now we are all knew that the male is the one who can control the type or sex of the fetus. Let's discuss the first point together. Any cell in our human body contains 46 chromosomes, which equal 23 pairs of chromosomes. These 23 pairs of chromosomes can be differentiated into two types, which are somatic chromosomes and sex chromosomes. Somatic chromosomes, also we can call them autosomes. They are 44 chromosomes or 22 pairs of chromosomes, and they are homologous pairs. What is the meaning of homologous pairs? They are similar to each others and similar in both male and female. The second type is sex chromosomes. Sex chromosomes equal two chromosomes or also one pair of chromosomes, and they are different between male and female. In female, they are similar to each others and they are XX, and also we can call them homozygous pairs. What is the meaning of homozygous? Homozygous means that they are similar to each others. In male, they are different to each others and they are XY, and also we can call them heterozygous pair. Now, let's discuss the main point, which says that the male is the one who can control the sex or type of the fetus. The male genetic structure is 44 plus XY, and the female genetic structure is 44 plus XX. According to chromosomal theory, chromosomes, when assorted in gametes, behave independently, so the male can produce two different types of gametes, which are 22 plus X and 22 plus Y. But the female can produce only one type of gamete, which equal 22 plus X. They are similar. All the female gametes are similar and their genetic structure is 22 plus X. So the male is the one who can control the type or sex of the fetus. According to it can produce two different type of gametes. Now let's discuss the second point in our lesson today, which is the abnormal genetic cases. But before that, we should explain the next example to can get our point. Let's see this example. The male genetic structure is 44 plus XY, and the female genetic structure is 44 plus XX. According to chromosomal theory, chromosomes, when assorted in gametes, behave independently. So the male gametes will be 22 plus X and 22 plus Y. But here in the female, an abnormal case will occur. According to these abnormal genetic cases, the sex chromosomes will assort in only one type of gamete, and the other type of gamete will be empty. So the 44 plus XX will produce an abnormal ovum. Its genetic structure is 22 plus XX, and another type of abnormal ovum, which its genetic structure is 22 plus 0. Let's see the probabilities which can produce from these abnormal cases. If 22 plus X, the sperm, will fertilize the abnormal ovum, which its genetic structure is 22 plus XX, the produced person will be 44 plus XXX, triple X, and this is called the polyploidy. What is meaning of polyploidy? Polyploidy means multiplication in the sex chromosomes, and this person will die. The second probability, when 22 plus Y, the normal sperm fertilized the abnormal ovum, which its genetic structure is 22 plus 0. The produced person will be male, and its genetic structure is 44 plus Y, and this is male will die too. Why? Because we can't live without X chromosome, but we can live without Y chromosomes, as all females. All females don't contain Y chromosomes because their genetic structure is 44 plus XX. Let's see the two other types of the abnormal genetic cases. If the normal sperm, which its genetic structure is 22 plus Y, will fertilize the abnormal ovum, which its genetic structure is 22 plus XX, the produced person will be 44 plus XXY. And this is one of the syndromes we are going to study today, which is Klinefelter syndrome. The last probability which can produce from these cases when the normal sperm 22 plus X will fertilize the abnormal ovum, which its genetic structure is 22 plus 0. The produced person will be female, and its genetic structure is 44 plus x0, and this is the other syndrome which we are going to study today, which is Turner syndrome. Now, let's study our abnormal chromosomal cases in humans. The first case we have just mentioned is Klinefelter syndrome. This syndrome was discovered by Dr. Henry Klinefelter in 1942. The causes of Klinefelter syndrome, as we have just mentioned, is fertilization of abnormal ovum, which its genetic structure is 22 plus XX instead of 22 plus X, by normal sperm, which its genetic structure is 22 plus Y. The produced person will be 44 plus XXY instead of 44 plus XY. So it contains an extra X. This syndrome appears in males only, and the symptoms of this syndrome is infertile male due to the absence of the cells which produce sperm. To mental retardation. What is the meaning of mental retardations? Means dysfunction in the development of nervous system. 3. The appearance of feminine characters like enlargement of breasts, tall stature, and the growth of limbs more than average rate. And this is the karyotype of Klinefelter syndrome. C. The last chromosomes, which is the pair in the 23, which is the pair which is responsible for determination of sex. Its structure is XXY. And this is the first case we are going to study today in the abnormal genetic structures. The second case is Turner syndrome that we have just mentioned, which can produce from fertilization from between an abnormal ovum, which its genetic structure is 22 plus 0, with normal sperm, which its genetic structure is 22 plus X, so the produced person will be 44 plus X0. And this syndrome appears in females only. The symptoms of Turner's syndrome is short statures. Woman doesn't reach puberty due to the leakage of hormones. 3. Slow mental development or mental retardation, and congenital disorder of heart and kidneys. What is the meaning of congenitals? Means abnormalities in the structure of these organs. Let's see the karyotype of Turner's syndrome. See, the 6 chromosomes, which is the 23 pairs, it contains only one X chromosomes, and this is the karyotype of Turner's syndrome. Now, let's study the last abnormal case genetic case in our lesson today, which is Down syndrome or mangulism. Mangulism, the abnormalities here not in sex chromosomes, but the abnormalities here in somatic chromosomes or autosomes, especially the chromosomes number 21. The chromosomes number 21 bears not assorted independently in different gametes, but here will assort in only one gamete. So we are going to have an abnormal ovum or an abnormal sperm which contains double copies of chromosome number 21. So the abnormal gamete, its genetic structure will be 23 instead of 22 plus X or plus Y. So the abnormality here, not in ovum like the other cases, Klinefelter and Turner syndrome, but the abnormalities here will be in the autosomes and may occur in both ovum and sperms. Let's discuss this case. Mangulism was discovered by British doctor Down in 1866. Causes of Down syndrome. As we have just mentioned, it caused by fertilization of an abnormal ovum or abnormal sperm, which is their genetic structure are 23 plus Y or 23 plus X with another normal gamete. The produced person, if female, its genetic structure will be 45 plus XX. If male, its genetic structure will be 45 plus XY. So the produced person will have triple copies of chromosome number 21 in karyotype. Now, let's see the symptoms of Down syndrome. Delayed growth, short stature, oval face, also short fingers and toes, mental retardation, narrow eyes, small ears. Let's see the karyotype of Down syndrome. See, dear students, it contains triple 21 chromosome in karyotype. So it has 3 copies of chromosomes in chromosome number 21 in karyotype. Dear students, I hope you understand our lesson today well. See you next time, inshallah, your biology teacher, Ines Adam. Thank you.